Hello everyone. In today's tutorial we will talk about random forest test using R packages. And what is actually random forest? Random forest is a supervised learning algorithm which is built using ensemble of decision trees and basically it tests and trains the data and find the best model for your variables. So let's start and perform the analysis using R. So basically I'll use a file, not this one, is for cancer dataset, this one. Here we have, let's see, here we have number of different gene information. For example, here this is gene 1, gene 2. If I just zoom in for your ease, that's gene 1, then this is the expression information of genes, then this gene 2, and this is the expression information of genes in these samples. And these genes are actually the variables or risk factors, or you can say explanatory variables. And now we will check the expression of these genes against any response variable, for instance age, stage, grade, metastasis, and so on and so forth. And so let's start and begin the analysis. So here what we will do is we will select all these genes, and then we will use one response variable and see what is the best model, or what are the best explanatory variables modeling against any specific response variable. So let's start here, right? So for first of all, what you need to do is to call the file, this file. Let's say I say it cancer, and I read .csv file, and this is actually present in E, videos, yeah, OBS, then R, tutorials, and then it's a cancer dataset, right? And then if you want to make it explain, if you want to explain the whole file, then you write head equals to true, then separate by this is CSV file, so separate it by comma, right? So just run this file and you can see this is the file I was showing you, genes and then the information, right? So next is, yeah, so now what you need to do is to call two libraries I will use here. Number one is dplyr package for selecting the columns, right? And the other one is the random forest. So these two will be selected here, so you just run those. The next thing is, it's very simple. For instance, if I want to say that this is the gene and attributes are the attributes, for instance, means I want to say that the response factors, I'm calling them attributes, let's say. And as you can see that there are some files which have NAs like this, and I just want to exclude those. So for that I just write gene attributes, then NA omit, this NA omit, and then I select a subset containing the cancer dataset, and I want to select some of the features out of all. For example, here I am saying that I want to do it for grade, tumor grade, and then gene 1, gene 2, gene 3, gene 9, and gene 10, right? So let's see if it's running fine, the code is fine or not. Yeah, so it's now, if you want to see the gene attribute file, you can just click and see here that now it has selected these genes and the grade information, right? Gene. If you want to see the first few lines, you can just write head and this one, and you can see the grade, gene 1, gene 2, gene 3, gene 9 and gene 10. Now what we need to do is to apply the grade versus genes. Let's see, and call the random forest test, and just write random forest as dot factor. I want to call this factor as grade, and then after grade, let's see, gene 1 plus gene 2 plus gene 3 plus gene 9 plus gene 10, right? And then from which data set? The data is data equals to gene attribute clean, right? And number of trees I want to generate, you know that it builds a lot of trees. I can just set it to n tree equals to 500, right? So now you can just run this command, and now the random forest test is almost done. What we have to do is to check the importance of each variable in the tree, right? So just write this one, and then you can see the importance of the tree, that the maximum value, the highest the value is for the best gene. So here the highest value is, all of the genes show good values and they are slightly closer to each other, but the maximum is gene 10. So if we just build the plot, you can see that if you just build the plot, you can see that the highest value is for the gene 10, and it will be on the maximum side. So here you can see that the maximum value is for gene 10, that is near to 46.43, and the minimum value is for gene 9, that is 43, right? So using this random forest test, you can find the best gene which is modeled, any feature you want to check with any other response variable, so you can do it very easily using R Studio and the random forest package. Thank you very much. I think it's enough for today. Bye bye.